So, if you look at my tie, you're going to see a lot of biotechnology. And what the biotechnology is doing for us now is revolutionary. Medicine, and something else, genealogy. Whether you're talking about the genealogy of one person finding out where their ancestors come from, or going way back to pre-humans. And but I want to talk about the genetic testing and what we use for genealogy. And there's a couple different companies, and I've done both tests. So I want to, before I do my results, I want to talk about the terminology used. And this is part of what I'm calling my DNA journey. Since I spit in a couple months ago, I have had the results, and I wanted to do a video on the terminology. Then I'll go over my results. Brought to you by Curious Randland, where science literacy will make America great. Hey, look, this is my tie. All right, so I'm also going to do this, and I'm kind of watching the time, and then I might go and do a little comparison between these two companies. These are the two that I've done. So I've already done a video on collecting the data for 23andMe and for Ancestry, and I'm putting it on my personal YouTube and also my Curious Randland one. So let's talk about the terminology. What makes us able to make these conclusions? And here's a variety of terms, and I'm going to address a lot of these. Let's start with this one. First, we've got 246 million nucleotide bases just in our chromosome number 1. And remember, DNA is confined, and a gene is a segment of DNA that codes for a protein, for a trait. And we have 23 pairs of chromosomes. If you're a male, you have 1 through 22. If you're a female, you have 1 through 22. The difference is, if you're a male, you have an X and a Y, and if you're female, you have an X and an X, 2 X's. And so those are called sex chromosomes versus autosomes. Now, the autosomes is how we will use this one called SNPs. All right, so let's walk through. Let's talk about the SNP. A single nucleotide polymorphism. Some people call it a single nucleotide polymorphism mutation. See, there's 23,000 genes, and each gene is made up of hundreds of nucleotides. We have 10 million variants. Again, our chromosome number 1 has 246 million bases. So why look at all those ones that are the same? Let's look at the differences. And what is this helping us do? It helps us recognize patterns where we see the letters are different. See, I might have on the a loci, that's called a location on a chromosome. I might have like 3 A's and then 3 T's or something like that. Someone else might have 3 A's, but then they'll have like 2 T's and a G. Or so it might be a single nucleotide that's different, and those are what we call variants. So what is the difference between genotyping and genome sequencing? Well, genotyping is looking for the variants, the variations, the SNPs. Genome sequencing is lining up, here's the first base to the end of it, and going the complete sequence. And that's incredibly expensive. But what we're doing is we're doing what's called genotyping. And that's what helps us find DNA fingerprinting, and it helps us do the test for like crime scenes and also for genealogy. And here's autosomal chromosomes. That chromosomes 1 through 22. It's also called our nuclear DNA. So what is a haplogroup? There's actually 2 areas. One has to do with male versus female. Everyone has the maternal one, and only half the population have the paternal. So we'll elaborate. So we got a similar haplotypes. Basically, they share, if they have similar SNPs, they have similar ancestry. What does that mean? We can find, what we've discovered is certain ethnic groups or certain geographic areas over time, which is what a haplogroup is. There's no such thing as race. There's, race is a human construct. So culture and identity in a tribe living in a certain area for a long period of time, that's a haplogroup. And we've discovered that each haplogroup has a unique SNP signature, so to speak. View this as a marker that helps us say, all right, this person, we can trace their ancestry goes to this continent or this continent. All right. So the haplogroup, you notice it says the Y chromosome and the mitochondrial. So these two make up the 2 different haplotypes. All right, and 23andMe doesn't actually use SNP. They call it variants. So, and here's, if you look at it, this is from their website. So they say one person might have an A at a spot and another person might have a T. This is a variant that we use to associate with disease traits and ancestral groups. All right? Because you get it from both parents, you get one copy from each of your autosomal chromosomes. So what are the haplogroups? The maternal, well, did you know that you have another type of DNA that's not part of your 23 chromosomes? There's an evolutionary relationship with this mitochondrial DNA. See, we have, we're what we call eukaryotic cells. We have different organelles. We got a nucleus, we got a mitochondria, which is our energy producing area. However, that mitochondria has its own DNA. And because it doesn't interact with your other chromosomes, with your other DNA, what we call your autosomal or your nuclear DNA, it doesn't interact with it. See, if this is 2 chromosomes, let's say this is chromosome 5, and they're going to basically exchange a piece before they separate during meiosis. Well, the mitochondrial DNA, they don't do that thing, and it's called crossing over. Because of that, there's less chance for mutations. Because there's less chance of mutations, the mutations jump out at certain time periods, at locations, so we can actually trace, here's a mutation, and it happened in this area, and it helps us trace our genealogical timeline. Okay, I said genealogical, all right? So let's move on. Here's a human mitochondrial, that's often pronounced mtDNA, and these are some of the different haplogroups. They use a letter designation, so one might be a haplogroup L, one might be a haplogroup R. Matter of fact, L is the original coming out of Africa. As they branched out, subtle mutations in their autosomal DNA, but their mitochondrial help place them in a different group because of their geography and their interactions. That geography and that isolation in a group creates a group that interacts with another group. Some call it race, but it's actually haplogroups, because they're all the same species. There's only one skin color, melanin. Right, so let's move on. The paternal haplogroup is isolated on the Y chromosome. See, the Y chromosome doesn't do crossing over. The X chromosome is not a good source for this because, like all the autosomes, it does exchange information. There's an element of crossing over, so there's more mutation, more shuffling of the genetic deck. It's harder to trace that. The mitochondrial and the Y chromosome, less mutation, so it's easier to find when there's been a major change. And we've seen certain markers show up in certain populations at a certain time. You're looking at migratory patterns. All right, so here's the paternal haplogroups. And all right, so a lot of times we call these genetic markers. And they're basically 2 alleles. They're for the same trait, and there's a subtle variation. Darwin said there's variation in species, and now what we know that we can find, like one person has an A at that spot, another person has a T, that SNP that we talked about earlier. All right, lastly, Neanderthals. According to 23andMe, there are 22,872 Neanderthal variants. Okay, so we did interbreed with Neanderthals, and the evidence is in our genes. If you have a dominant European background, you're going to have more Neanderthal variants. If you're not, you'll have less. All right? So, of all the population in the database, and one of the things I need to mention is the database, as we get more and more people who are doing these tests, we broaden. We sometimes have to redefine the locations. Maybe one time they can say, all right, we can trace you to the Rift Valley, the Great Rift Valley. Now we're saying we can trace you at a certain part in that Rift Valley. Okay? And so the more we have, the more data we're going to be able to redefine. So even with Neanderthals. So with Neanderthals, you'll notice of the 23andMe's, the person that has the most has been 397. All right, so let's talk about the 2 different softwares that I'm going to use, or that I have used. One's 23andMe and one's Ancestry. So this is not my full result. This is just a snapshot of my results. You can see my percentages. I'm not going to talk about it now. I'm going to do a video on this and a video on that. Here's Ancestry. They gave me a little part. And I've only found this once. I did a screenshot, and every time I log on, I can't seem to find this. And I would click the full results. So the look of the 2, when you go to 23andMe, when you click reports, this is where you're going to follow, and it gives you ancestry composition. 23 gives you the maternal and the paternal haplogroup, and they tell you about your Neanderthal ancestry. All right. Ancestry doesn't give you the haplogroups, but their database, they can trace back to geography going back to 2,000 years. So for 23andMe, if you want the long term, like with one of the haplogroups we say originated 78,000 years ago in a certain part of Africa. That's not, Ancestry won't be able to do that, but they'll be able to go back and look, think about what the history of what was going on 2,000 years ago, and that's what they can do. So you can find your genetic communities and everything. So I'm going to do a separate video on each one of these results, and just make sure got everything. All right, I've got some great sources. I'll put these in the description. And there we go. Brought to you by Curious Brandland. So what I'm going to show next is my 23andMe results and then my Ancestry. And one last thing I want to point out is all of these videos are in a playlist that I've called Ancestry and Genealogy on my Curious Brandland or my personal one. There's two really good videos here. These three, actually three. These are not my videos, but she goes into the details of how they come up with the SNP and how they do that. This guy goes into the techniques of the actual, if you want to know about how they actually match up and find base pairing. And this lady does a really good comparison between 23andMe and Ancestry. And there's like 2 or 3 more others that are pretty good as well. So that's my DNA journey, the prelude to my actually going over my results. So now I'm going to do a video on my results for 23andMe and Ancestry. Because 23andMe goes into more details with ancestry composition, so it's chromosomal, that'll be a longer video. The Ancestry will probably only be about 2 or 3 minutes. So again, thanks for watching and following me on my journey. So I'm going to conclude what I did with my other ones. What we are is determined at birth. What we become takes a lifetime.